Hi everyone. So today I'm going to be talking about my research interest, which is drug delivery, and specifically nanoparticles, and even more specifically nanomedicine, and why I think that they're one of the best solutions for current therapeutics. So to really understand the significance of nanomedicine, we first need to understand what nanoparticles are. So as expected, they're defined as particles ranging from the size of 1 nanometer to 1,000 nanometers. So this is significant because when you look at the figure, you can see that eukaryotic cells range in the size of microns, and specifically the biological targets that are pertinent for research, such as nucleic acids, proteins, and viruses, all fall within the nanometer range. So having a particle in that size is key. Another unique property of nanoscale particles is that we're able to change its physical properties with just slight changes in the nanoscale sizes, such as melting points, optical absorption, thermodynamic properties, and so forth. And we know that the surface charge ratio to the volume ratio is really important in loading. This helps increase the permeability, and thus you can reduce your dose. Therefore, size, shape, and surface are really important. So there are 5 main categories of nanoparticles that we can see in the figure: lipid-based, polymer-based, inorganic, viral, and drug conjugates. So instead of getting specifics of all of these, I'm just going to highlight a few. So liposomes are spear-shaped, and they usually have at least one phospholipid bilayer. The unique thing about liposomes is that you're able to encapsulate both hydrophobic and hydrophilic drugs. So the hydrophilic would go into the aqueous core and dissolve, whereas the hydrophobic could be on the phospholipid bilayer. Additionally, you can encapsulate the drug. Usually cancerous drugs are cytotoxic, so being able to encapsulate it and control its release is really important in making sure it gets to its target and doesn't kill other cells along the way. Another category is drug conjugates, which is made up of a monoclonal antibody which can bind to a specific receptor, and it's linked to a cytotoxic agent. So there are already 5 approved drugs for metastasized cancer in the drug conjugate category. Lastly, there are metal nanoparticles, which can be used for imaging or in its ability to kill bacteria, such as magnesium oxide nanoparticles. A little bit of history about nanomedicine. These aren't a really new concept. The first liposome you can see was created in the 80s, and its first approved nano construct was approved in the 90s. Lately, the push has been to be able to encapsulate mRNA drugs into a nano construct, which we can see towards the right. So the advantages of nanoscale medicine in a research context, we can see that there is a push towards RNA drugs being encapsulated, and this is because one of the key pros of nanoscale medicine is being able to co-deliver therapies. So you can see that you can co-deliver chemotherapeutic with an RNAi therapy or even a theranostic imaging agent. We're able to target the delivery to the disease site using ligand-mediated targeting, and this is important for transcytosis. Also, you're able to increase the bioavailability and solubility, which means increased uptake of your nanoparticle or whatever your drug is by endocytosis. So one example where a nanoparticle increased the efficiency of a agent is doxorubicin. So doxorubicin is a cytotoxic agent that inhibits topoisomerase II, which we learned about in class. One of the issues is that there was a lot of cytotoxic free radicals in the body, resulting in cardiotoxicity and death. So clearly there is a need for increased specificity, and this was resolved by Doxil, which is a single-layer liposome. It's shown to reduce cardiotoxicity and also have a longer elimination half-time and a longer survival rate in patients. So since we can see there is such an improved outcome scientifically of delivering agents in the nano construct, why are we not seeing it for all our medicines? So one of the biggest issues is manufacturing it. So a lot of steps, a lot of vessels, and a lot of time to be able to get the specific purity, the binding affinities, and the biocompatibility that we need out of the drugs. Here are some of the references I used, and thanks for listening.